Welcome to Fluid and Electrolyte Mastery, a concise framework for how kidneys keep our internal world in balance. In this presentation, we'll trace how water and ions move, how sensors and hormones respond, and how those processes translate into clinical problems and treatments. Using the diagrams here as a roadmap to guide our understanding of renal regulation. This slide breaks down total body water into intracellular and extracellular compartments and highlights the forces driving fluid shifts. The diagram shows how osmotic gradients and Starling forces move water between plasma and interstitium, while the table summarizes typical volumes and dominant ions, giving you a practical sense of where sodium, potassium, and chloride primarily reside. Glomerular filtration is the starting point for renal regulation, producing roughly 180 liters of ultrafiltrate daily. The central graphic shows how high renal blood flow and capillary hydrostatic pressure drive filtration, while the selective filtration barrier limits passage by size and charge, setting the stage for downstream tubular reabsorption and secretion that ultimately tailor urine to bodily needs. This slide maps nephron segments to their transport roles and the key transporters that determine urine concentration. The illustration links proximal reabsorption, the loop of Henle's countercurrent multiplication, and distal and collecting duct fine-tuning by ENaC and aquaporins, emphasizing how cumulative transporter activity and ADH action shape final urine composition and systemic water balance. Sodium is the principal extracellular cation that sets plasma osmolality and effective circulating volume, and the slide shows how RAAS and aldosterone modulate sodium handling. Aldosterone upregulates ENase in the distal nephron to increase sodium reabsorption, expanding volume, and influencing blood pressure through tightly regulated feedback loops between kidney sensors and hormonal systems. When volume expands, the body activates renal baroreceptors, macula densa signaling, and atrial natriuretic peptide to suppress renin and aldosterone, as shown in the flowchart. These coordinated sensors inhibit RAAS, increase natriuresis and diuresis, and restore blood volume toward normal, preventing fluid overload and protecting the circulation from sustained pressure elevation. Vasopressin, or ADH, is released when hypothalamic osmoreceptors detect hyperosmolality, then acts on V2 receptors in the collecting duct to trigger a CAMP cascade. The diagram traces AQP2 vesicle insertion into the apical membrane, increasing water reabsorption, concentrating urine and diluting plasma osmolality to reestablish electrolyte and fluid balance. Thirst provides a behavioral complement to hormonal regulation. Hypothalamic osmoreceptors stimulate a conscious drive to drink when osmolality rises, and the resulting water intake reduces osmolality and suppresses thirst through negative feedback. This slide highlights how drinking and renal ADH action synergize to normalize body water without overcorrection. Principal cells in the collecting duct coordinate potassium and sodium balance by coupling basolateral NAKAT pace with apical K channels like ROMK and BK and the ENaC sodium channel. Aldosterone amplifies this system, increasing Na reabsorption and K secretion. And the slide reminds us that dysregulation can cause dangerous hyperkalemia or hypokalemia with major cardiac and neuromuscular consequences. In metabolic acidosis, the kidney compensates through proximal bicarbonate reclamation, increased ammoniogenesis, and enhanced titratable acid excretion, as illustrated here. The HKAT pace also shifts to conserve potassium during acidosis, and the panel on the right summarizes reciprocal electrolyte effects. Acidosis tends to raise plasma K and Ca, while alkalosis has the opposite impact. This clinical mapping links common presentations to specific nephron transport defects. SIADH reflects excess ADH and dilutional hyponatremia. Hyperaldosteronism drives hypokalemic alkalosis via ENaC and NAK, ATPase activity. RTA corresponds to impaired H+ secretion, or bicarbonate handling, and diabetes insipidus results from deficient ADH action with hypernatremic dehydration, tying symptoms to mechanisms. The final slide offers renal-guided interventions, restore perfusion, and give graded isotonic saline. For hypovolemic hyponatremia, consider vasopressin antagonists to increase free water clearance in euvolemic SIADH. Use potassium binders to reduce systemic K+ burden in hyperkalemia, and always correct free water carefully to avoid osmotic demyelinations while monitoring closely.